Recording. So you're taking notes. I'll collect the safety contract at the end of the period or near the end of the period. This block ends at 10:48. It is going to be 10 o'clock now, so we have 48 minutes. If it were up to you, we'd only have 2 minutes because you'd waste all your time talking while I'm talking. All right. Anyways. So yes, you're taking notes every day. No. What?
Are we taking notes?
I don't know why I'm answering these questions. It boggles the mind. But let me be clear. Expectations. For AP. I think they're reasonable. They're pretty straightforward. This is what happens in every class. So I don't know why hasn't sunk in to at your grade level. But number one, you should be reading chapter 2. You should have already uploaded. If you haven't done so, I think today's the last day for you to do it to get any credit for it. You should have already uploaded chapter 1 notes. If you haven't uploaded chapter 1 notes, then it's going to be a 0 after today. Should be reading chapter 2 and taking notes. But to save you time, and when I say taking notes for your class, I'm trying to save you time because you have a lot to do. That means do the study guide. Correct?
Yeah.
So you have a study guide for chapter 2. I expect you guys are reading. If you're just doing a word search and you're not reading, you're missing a good part of the content. You're hurting yourselves. But there's a limit to how much I can chase after you. So read chapter 2 and then fill in the study guide. You have it in front of you. I printed it. It's also posted. The second thing you should be doing is getting ready for Thursday's lab. You have a lab you have to do. You're going to use this equipment. You're not creating a terrarium with half a 2-liter bottle like they ask you to do in the written part of that lab. In fact, the one I wanted to photocopy was one where I whited out those particular directions. I was going to have you design your own labs, and that's why you have your whiteboards and expo markers in front of you. You're going to work in groups today of 4 or 5, and you're going to design an experiment. Your experiment is going to have to do with salt solutions. It's going to have to do with you walking through how do I test the impact of salt on plant growth. So you should be doing that. And for this, you should be taking notes, not texting. What I would expect is you have a pencil in you're taking notes on your phone?
Yeah.
You know I'm collecting a notebook.
No. This is like my notes like on my to-do list.
I see. So I'm collecting a notebook. I expect to see Cornell notes. So my ideal would be page, title, main ideas, details, summary. That's what I expect to see. Writing in your phones and in your computers and all that, do what you got to do. But I need to see a notebook in 3-ring binder, everything placed in order by date. Punch holes and stuff that don't have it. Not a spiral notebook. Okay, I'm just telling you. That's what I'm going to be expecting when I collect notes in about a week and a half, maybe 2 weeks, because I understand you guys have some senior things that are going on. Yeah.
If you take notes like on loose leaf and you keep it in the binder, is that
That's perfect.
Oh, okay.
That's what should be happening. That's what should be happening. But I see a lot of stuff going on around me, and I just want to make it clear. I shouldn't have to say this to you. To have to do this all day with ninth graders. I expected. At your age, I expect you listen the first time. I'm not talking to anyone specific. I'm just saying bottom line. That's what we have to do. So ready for Thursday's lab. And to do that, you have to understand, you should have already skimmed through the lab that I handed you yesterday, and you should have some idea what we're doing. So you're gonna, instead of doing the terrarium where you're cutting the 2-liter bottle in half and creating water, and what they want you to do is to set up a 2-liter bottle like this and put one inside the other. And let me do a separate color. And put one inside the other. And in this one up here, you would put earth, and then here you would fill it up with water. And there's holes in this. They will actually want you to have a wick, so it wicks the water up, and then they want you to plant a seed here. And of course, this water is going to be different. So there's going to be a different water in this that allows it to wake up. That requires less watering on a daily basis. So, okay, we're not going to do that. We're not going to end up creating these little terrariums. You'd have to make one a piece, and there'd have to be a lot of 2-liter bottles being used, and etc. So we're not doing that. So forget what they said as far as that's concerned. That's part of your design. You're changing that design. What you're going to have are these little planters. You're used to them. They're right over there. There's little black planters. You're going to make 5 planters per group. All right, I'm sorry. Let's not use group. Per team. Team of 4 to 5 students. You're going to have 5 little pots. You're going to put in soil into each pot. The same soil. The same soil. You're going to put one seed, one tiny little seed into each pot, and you're going to have 5 different solutions. So you have these beakers. Actually, it probably might be better use an Erlenmeyer flask. And there's going to be 4 different ones. And 5 different flasks for each of the 5 different conditions. You're going to have 4 different experimental groups. And you're going to have 1 group that's not an experimental group. You're going to tell me what that is. So I believe what they have for you is you're going to have a 0.5% salt solution, a 1% salt solution, 2.5% salt solution, and a 5% salt solution. And you're going to use this salt solution to water this one. You're going to use this salt solution to water that one. This salt solution you're going to water this one, and this salt solution you're going to use to water that one. What are these called? Who knows what this group is called? Yeah.
The experimental group.
It's an experimental group. This is what you're using, what you're changing. What variable, what is A? The variable that you're changing, what is that, by the way, what is the one that you're changing? What do we call it? Independent variable. And so the independent variable is equal to salt concentration. Do you agree? I'm using square brackets to mean salt concentration. So what is this one over here about? What is this?
The control.
The control. I shouldn't use blue. This one over here is the control. And what is this one? What percent? What am I doing with the control?
0%.
0% is correct. So this obviously wants 0% salt, this distilled water. Now why do we put in distilled water? Why are we adding distilled water to this last plant when we're trying to see what the relationship is between the salt concentration, A, and B, the growth of the plant? Why are we doing that? Why would we bother with a zero? There's no salt in it. What do we do with a controller? What do we do with a controller?
We compare.
Compare. That's right, we're comparing. We do this because this is what we call our positive control. We expect this plant we're going to water it exactly the same volume of water is going to go to each plant at exactly the same time. I have to. I just have to.
Oh, snap.
Give it to me.
It's for the thing.
What is it?
I promise I don't have my glasses, so I'm on my
using her
camera.
phone to take photos because she can't see the board.
Oh.
Scared me like that.
What? How am I scaring you? I just said give me the phone.
Yeah, because I was confused because I'm literally
Well, you know you can grab your stuff and go sit right there, right in front of the
That's not
screen.
going to change anything. I'm still not going to be able to see.
You want me to get you some pop bottles?
My glasses at home. I'm going to be cool tomorrow. I just can't
Binoculars? No. All right,
see. If you got something,
because I have bad eyesight and I can see sitting right there.
well, according to my eye doctor, my eyesight's terrible. So
Mine's terrible too.
Not gonna talk about eyesight.
I'm just trying to give her a solution to the problem.
You can ask Shark. I know.
Oh my God, all right. Anyways, control, a 0% solution is here we're gonna put the same volume of water in each one. So you're keeping the volume of water the same. The light in these light tables, what you're gonna do is you're gonna have these rectangular lights, they're gonna,
UV light?
they're regular light, grow lights, they're sunlight bulbs. I might have to buy a couple, I don't know. And underneath those, there are trays. You can see that, right? That underneath them, there's these trays that sit like this. And in those, you're going to put your little things of soil. With your one seed. And those planters, those little black planters have small holes in them. And each tray is going to hold a certain amount of liquid. So we'll make sure that we have water down here. for the weekend, because we won't be here to water the plants over the weekend, we'll fill up the water here in the trays so that the water will wick up if necessary. And again, each tray will hold a different amount percent. One tray will be 0.5, and there'll be another tray that's 1. There's 1, 2, 3, 4, and then the fifth one will be the clear water. So 5 trays, 5 sources of water. And that's the setup you're going to use to do your experimental design. Is that clear to everybody? You're going to collect data. Now, what is this? There's an option B as well. You're going to do both options. You're doing option A and this is option A. This is going to take about a week to get done. It'll start to grow. You'll have all the data collected for option A in a week. But option B, which you'll have to continue to do, is just keep it growing. The ones that are growing are going to keep growing. You have to label these, by the way, when you get your planter. You're going to get some masking tape. You're going to label them with your team name. All right, on the outside. These seeds are marigolds. And do you know what marigolds are?
They're flowers. You usually like yellow.
They're plants. Yeah, they're yellow and orange. Everybody see? You want to go get the pictures? Go get them. They're over there, the little packets of marigold. I have 1,000 seeds plus these, so there's plenty of seeds. You're going to kill a bunch of them, obviously. I hope it's obvious. Yeah, salt solution is going to kill them. But there's going to be some that survive. Yeah. This is what they look like. They're different colors. And so you're going to take them and you're going to grow them. You're going to continue to grow them for option B. The seeds are actually weird looking. They look like little spirals, and you'll see why in a minute. Or not in a minute, but when you look at the flower anatomy. So these plants are going to grow, and they're also going to flower, and they flower into these really big. You saw what they look like, right? Like they're big and radial, and they get colorful. They're orange or red or yellow. So I should just use some yellow here. Yellow and so this is an interesting flower, marigolds, and that's why they call them marigolds, because they tend to be goldish yellow. But you should know that marigolds are not single flowers. They are actually what we call compound flowers. So one of the objectives in B is to pollinate these flowers. So what you're going to do, you're going to cross-pollinate them. You're going to take one compound flower, you're going to take a little brush, and you're going to act like a bee and go into a single flower, which is going to be not easy. And you're going to take up the pollen, you're going to carefully transfer it over to another flower, and that's going to create a seed where you're crossing the genes, right? It's called cross-pollination. You're going to act like the bee, and you're going to help these flowers do what they need to do. You should understand what I mean by compound flower is that it looks like one flower, but actually it's made of these tiny little flowers that look like this, and each one's a different color. That's why you can have multicolors in that. There's actually 2 kinds. I'll have you watch a video on it. And some are kind of a little janky thing. It looks like this. And there's little rings of this. And then the flower almost I think it looks a lot like in some ways looks like a sunflower, but not. All right, so anyways, it's not a straight stem. It has this little pocket. Looks like that. A little bulge. And you're going to dissect this and you're going to look at the flower, and what you're going to do, there's a dissection microscope, and you're going to be able to see that inside here are the fruits. So there's seeds, fruits of the marigold. So that's not the goal, but I thought you might be interested in seeing identifying the stamens and the ovaries and all the things that I told you the flowers have as sexual organs of the plant. The goal here, though, is to cross-pollinate them and get some idea of what someone like Mendel did. All right, that would be B, and you can look at B individually as you wish, as you read. And now you're going to design. You're going to spend the rest of today working on these easels. You have both a group objective, and you have an individual objective. And in your group objective, you need to read, and you need to design your lab. And by design, I mean I need to know that you understand what are your experimental groups and what are your control groups. And if you have a control group, maybe you decide to use, I don't know, no water in one. I have no idea what you're going to decide to do. But pure water in another. But you need to have that all designed out. You have to, as a group, you have to come to a consensus on the answers to the questions and the answers to the labs. And then as an individual, you have to have everything copied, everything written in your own handwriting. And then that's number one. And number two, you can have your own answers. They might be different from the group's answers because you may not agree with the group. So the key here is to come to a consensus for the group answer. So when you're sitting there and you're asking yourself, well, what does consensus mean? That to me, consensus does not mean that you all agree, nor does it mean that you've compromised. So a compromise is Bart is arguing and the answer's A, Noah is arguing the answer's B. After a while, Bart just gives up and says, okay, just put B on the paper, I'm done. I'm okay. That's a compromise. That's not how you come to a consensus. In a consensus, the team has a discussion. You come to, it doesn't mean that Bart gets A and Noah gets, or Noah gets B. Maybe Bart convinces Noah, maybe Noah convinces Bart, or maybe you both write something that's in between A and B, right? But in a consensus, everybody in the group mostly agrees. The key is mostly agrees. You don't have to have 100% agreement. If you disagree, you have some other thing you would rather write or say, you can write that in your own individual answers. But as a group, you should have consensus that lab questions. You should know what you're doing for Thursday. You should have a to-do list listed out, all the equipment, all the personal safety equipment. I'll give you a hint. The only thing you need is gloves and an apron. You're only working with dirt and water and salt. So there's no real chemicals. You don't even need goggles unless you're afraid you're going to get salt water in your eyes. But there will be no food on Thursday. So even if you bring it yourself in a packet, prepackaged, and it's multicolored and full of sugar. So, are we all clear? Everybody good with that? All right, well, good luck to you. Now, wait, I'm going to assign groups now. I'm going to assign groups because, no. I'm going to assign groups because I know. I know. Here was my test. My question. I always ask myself that question. What should I just let the kid? They're old enough. Should just choose their own quiz. I would love to do that. I would love to be hands-off. All right, here's your task. See you guys later. I'll see you. I'm going to work. I'm going to put on my headphones and get my work done up here. You guys do your thing. Peace. Will that work? Don't tell me yes. No, it will not. Exhibit A. The work I already assigned you. 10 of you have not done it. How hard was it to answer the study guide questions and upload it? Not hard. But 10 people haven't done it. That's without, I don't know what to say about that. Exhibit B, throughout the last, what is it, we've been in this class for a week. Exhibit B is, I've had several different side conversations going on, and that's just me giving you instructions on how to do and you know the lessons we've had. Now I'm going to trust these 10 people to go ahead and work on in their groups and trust that the person they're assigned to is not going to do all the work and then they're going to copy it. Trust that they're going to have everything ready on time and not come up with an excuse that they couldn't get it done because there wasn't enough time. Where does that trust come from? What evidence can I use to develop that kind of trust? So, no, I have to assign groups. Chardon, why don't you go up and join those ladies there? One of you two, I don't care which one, come on over here. One of you, I'll go over and join them. Jessica, go ahead and join this group back here. You two move over here. Bart, join them back there. Noah, join this group. You, come on, get your stuff together and go over there. Go join Dysart. Yeah, that's fine. You join, no, you can stay there. You two stay there, so you're 4 so far. You move over with Dysart. I think that's reasonable. So that's 4, you go into 5. Go and join that. No, you're there, and you, join that group back up with Jessica. Are you in this group? It's 3, there's already 6 there, we have Maria, so there's 4 there, you join that group back there. Both of you go join Jessica's group. Since they got 5 here, why don't you join those ladies back there? That'll make it 4. Yeah, you join them. Join that group over there. I don't even it out. So we got 6 here, 5 there, 3, 4, 5 there, and then Sharon's with you guys. So 3, 4, 5. Wait, where were you? Oh, with them. All right, so you can join them as well. So that's 6 there. All right. So make sure you're working as a group. Make sure your names are placed in your group. And I want a group sheet turned in today on what you've done, what you've accomplished by end of period. So right now, end of period is, what is it? End of period is 10:48, and it is 10:26. So that gives you 20 minutes. 20 minutes to give me something of a beginning of your plan and what you're going to do on Thursday to accomplish those tasks. All right, good luck.